Welcome to the pan-genomic model, part 7. In the previous 6 sections, I've outlined the pan-genomic model. What I'd like to do here is to try and tie everything together. Perhaps the most important concept is that this is going to apply to all kingdoms. It's minimal yet comprehensive. It's based on the well-studied binding of the transcription machinery to promoters. And this is shared with many, many other models. A central structural motif is a loop attached to a factory. And these two features are going to ensure that higher order structures appear spontaneously without the requirement for any other mechanisms. Function is going to be irretrievably interconnected with structure. Transcription drives the organization, and the organization drives transcription. The firing frequency is going to depend on the distance of a promoter to an appropriate factory. Here, the red and the green promoters are tethered equally close to the pink factory, but only the red one is likely to fire often. And this is a differentiating feature of our model. And small world networks appear inevitably just by the presence of different appropriately colored factories. Now, I've focused on transcription, but because the transcribed genome is the substrate for all of the other things that happen with that genome, of course, the pan-genomic model is going to influence these additional functions too. I think the model is incredibly powerful. It explains many mysteries. I've summarized some. For example, how mysterious motifs like enhancers and EQTLs work. I've also managed to reconcile the apparently irreconcilable. Yamanaka's result says that gene regulation is simply controlled, but GWAS points to the opposite. I've provided you with a very simple equation that it can predict the firing frequency of any genic or non-genic promoter in the genome that depends upon only one variable. So I think you'll agree that it is a pan-genomic model because every base pair in the genome is influencing the activity of all others to some degree through these distance effects. Promoters are obviously going to play important roles, but then open euchromatin, closed heterochromatin, and loops anchored by cohesins are all going to affect how frequently promoters interact with their appropriately colored factories. Another key feature of this model is that active polymerases are transiently immobile, just whilst they're active. But despite the evidence for such immobility, recent reviews still assume that they track. So this is probably the question that I'm asked the most. Why don't people accept your model? And of course, it's difficult to find an answer to that, but maybe behavioral psychologists do have an explanation. And it's called theory-induced blindness, and summarized by this quotation. Once you have accepted a theory and used it as a tool in your thinking, it is extraordinarily difficult to notice its flaws. If you come upon an observation that does not seem to fit the model, you assume that there must be a perfectly good explanation that you are somehow missing. I hope I have opened your eyes and convinced you that this pan-genomic model explains the fascinating interrelations between structure and function. Throughout, I have given a high-level view. For more detail, see Section 8. Thank you for your interest, and especially for coming this far.